everyone. Good day. My name is Rafter Manuel from Lorma Colleges, College of Nursing. I am a fourth year student nurse. And so the topic that I will be presenting to you this time is, if you can see in your screen, the tip of the iceberg. I'll be talking about the major aspects of cancer cells. And so I hope you will learn something from this very short lecture. Okay, let's talk about cancer cells. Let us first define what cancer is. You see, the word cancer has been roaming around, floating for decades already. But what does it mean? The easiest definition that I can give you is that cancer is a disease. It is a type of tumor that becomes progressively invasive, which can, transfer from one region of your body to another region of your body, which can destroy cells and would rapidly divide the cells. So basically, we do have cells in our bodies. And there are so many of them. In fact, according to Ursula Barth of 2017, Barth stated that there are about 37.2 trillion cells in a human body. So in the normal progress of the body, these cells, they divide, and after some time, it will grow. You see, this divide and grow is regulated in different stages. And that is why the hormones in our body come to play, and we call them the growth hormones, which are responsible in cell growth. However, we need to remember that when the growth development of the cell is triggered in a hyper state, the cell will never stop dividing itself. Meaning to say, they will increase in number. Because of this, it will constantly divide, continuously divide and divide and divide to produce an immortal line of cells. It will make a pile of cells into a particular region of your body, and we call that a tumor. And when the tumor becomes invasively progressive, meaning to say it moves from one place to another in our body, it makes them cancerous. Normally, the cells in our body, they grow, they divide, and then eventually they die. But these hyper state triggered cells would not die, but they become immortal. They cannot receive any signal, and they will be present until death, which is the major problem of cancer. What makes it more of a major problem is that those cells cannot be killed because the so-called killing mechanism in our body cannot recognize those cells, which means it will thrive. It will spread throughout the body. It means to say that your very own cell, your very own system is against you, and you cannot protect it from going against your cell. Now, if you look at in your screen, the normal mechanism of our cell. This is the normal mechanism of our cell. Say for example, this is the cell. Okay? Inside the cell is the nucleus. Now, because of the growth regulators produced by your hormones in your body, they divide themselves. Okay? They will divide and then they will further divide. And that is the normal mechanism. After their division and after their growth, there is what we call the killing pathway or the killing pathways. Meaning to say, after their division, after their growth, they will be killed. Okay? But in cancerous cell, it is growing faultily. They are growing faultily. Why? Because growth regulation increases while killing mechanism decreases. So dumadami sila dito while the killing mechanism decreases. That is why the growing division in the cell and the killing division of the cell alters this normal cell process because they are faulty. Eventually, what will happen is that the cell is going to divide and divide and divide. They won't stop dividing and they won't be killed. Why? Because, you know, the process has been altered, okay? Which will result in a massive growth of the cell. This production is, this production of, the massive growth is what we call tumor cells, and it becomes immortal. So, the growth regulator increases and the killing mechanism decreases. But it doesn't stop there. Because they are immortal, they are also invasive, meaning to say they can penetrate to the small tissues of your body. It can move from one place to another. It can spread, and it is called malignant tumor cells. And we will go to detail about the malignant tumor cells in a bit later. But now let's talk about how these normal cells became abnormal. What went wrong? Ano ang nangyari? Something triggered in the nucleus region, here in the nucleus region, that caused the normal cell to become cancerous. There must be some change in the nuclear region because, you see, the whole system inside the cell is being controlled by the nucleus. Therefore, there must be some change in the chromosomes of the cells. So what are the various factors? Okay? What are the various factors of change in the chromosomes? Number one, let me share to you, the first various factor is random mutation. This is what we call random mutation. And it can be caused by two things, either chemically or physically. Chemical carcinogens such as iridium bromide. Physical, because of UV radiation. These things change, these things alters the nucleotide sequences in the DNA, which can be triggered, and these healthy cells will become unhealthy. Another factor is eating habit or environmental. It could be due to the infectious kind of agents that enters our system. Number three, viruses. Viruses can also change the chromosome sequence into something else that will trigger it to become a cancer cell. So the changes in the nucleus are related to chromosomes. They are related to DNAs. But these changes can also be inherited from generation to another generation. So the fourth factor that I want to share to you is inheritance. For adopting these changes from one generation to another generation, change has to happen in the germline cells instead of the somatic cells. If something happened in the somatic cell, it won't travel to the next generation. Why? Because it will die with our cell. But if something happened in the germline cell, if something happened in your sperm cell, in your egg cells, those types of mutation can be taken to the future generation, which can lead to the change. So the change has something to do with the DNA. Thus, changing the chromosomal segment of the DNA segment will change the protein production. Okay? As it changes the protein production, the further it changes the regulation of growth and the killing of the cell. And eventually, converts it to cancer cells. Now, this is the very first part of my lecture. If you cannot remember what I have said earlier, I would want you to remember this. I'd like you to remember the normal process of the cell. You see, the cell, they divide, okay? They divide, and then they will grow. But normally, after they grow, after they divide, they will be killed. Otherwise, they will pile up against each other until a certain region in your body will become a tumor. Okay? And the worst case scenario is that it will spread to other regions of your body and may cause the death of a person. And so we have talked about the introductory part of cancer cells. Let's now talk about the important properties of cancer cells. The important properties of cancer cells. What are the advantages of them having these certain properties? But I want to tell you this first, that these cancer cells, they are the bad guys. We are the good guys. We are the healthy cells. The cancer cells are the bad guys. They are the unhealthy cells. And so they have their own advantages. And if they have these certain properties, they have these certain cancer cell properties, it is an advantage for them, but it is a disadvantage in our part. So the very first important properties of cancer cells is what we call immortality. Because of the programmed cell death, normal death pathways in the cells will divide and they will be killed, okay? But in the cancer cells, there is no apoptosis, okay? Meaning to say, if there's no apoptosis, there is no programmed cell death present in our body. That is the first important properties of cancer cells. Secondly, the second important property of cancer cells is what we call the rapid growth and division. This rapid growth of the cell is obtained by signals as a form of hormones, which are chemical mediators. You see, inside our body, some systems control the growth and division of the cell. Now, what is the cause? The number one cause is faulty checkpoints. Now, if you see here in your screen, this is actually a checkpoint. G1, checkpoint G1, checkpoint S, checkpoint G2, checkpoint M. And the cell has to go through these different checkpoints, okay? There are checkpoints in between checkpoint G1. There's a checkpoint here between G1 and S and G2, G2 and M. Okay? During the cell growth and division, the cell would have to pass through these checkpoints. For example, the cell examines whether all the sister chromatids are correctly attached to the spindle microtubes, or if the cell size is correct, or there is a DNA damage, so on and so forth. So this is the very first cause of rapid growth and division, faulty checkpoints. Okay? But if these checkpoints fail to control the division, it will lead to rapid growth and division. For example, you see here the S checkpoint. This is the S phase or the synthesis. The chromosome duplication happens here. Now, if the duplication of chromosomes did not happen due to some certain reason, duplication of chromosomes is not sufficient. What happens now is S and G2 will block the cell to enter the G2 checkpoint phase, which will freeze the cell back to the S phase. And some chemical mediators will come and tell the cell, hey, you have not produced enough chromosomes. You need to go back. Kinakailangan mo bumalik. Okay? You need to produce it first and then you come back into the G2 phase. That is why this faulty checkpoint hinders cell division. Okay? What other things that involve rapid growth and division? Cause number two is overproduction of growth receptor. You see here, the cell has receptors in the cell surface. This is the normal production. Let's say this is the normal production of growth receptor. But over here is the overproduction of growth receptors. Napakaraming receptor. Okay? See, these cell receptors outside the cell will receive growth signals or growth hormones which will send chemical signals to the nucleus inside the cell and then cause the nucleus to produce proteins in which it will start making the cell and it will grow to a certain size and then it will then be divided. So normally, when a cell is near the death phase, it will it will reduce in producing cell receptors. But in cancer cells, as you can see here, there is a there is an overproduction of growth receptors. Therefore, the cell will continuously receive growth hormones and they will not stop growing in size. Another important property of cancer cells is what we call the no contact inhibition. No contact inhibition. So normally, when a cell is in contact with another cell, and this cell is in contact to another cell, and this cell is in contact with this cell, meaning to say they are overcrowding this petri dish. They are overcrowding this place. Okay? Again, normally because of the cell, the nature of the cell, they communicate with each other. That they should not divide. They should stop dividing themselves because they will overcrowd this place. Okay? But in the cancerous cell, even though they are already in contact with each other, they are not stopping to divide, which makes them which makes this petri dish overcrowded, which makes this region of the body overcrowded. So that is the no contact inhibition. The fourth important properties of cancer cells is proliferation. You see, the cancerous cells can multiply from one place to another, which makes them dangerous. Not just because they can move from this region of your body to this region of your body, but they also invade tissues and produce tumors in different regions of the body. This property is what we call metastasis. Now, if you can see in your screen, there is a tumor cell here, okay? Because of because of proliferation, the cancer cells would travel into the bloodstream and it makes them very dangerous. Why? Because they invade tissues. So these cancer cells got out of the bloodstream because of their nature. They invade tissues and so it metastasized. Okay? There is a new tumor growth outside or away from the original region. Another feature of this cancer cell is the low nutrient and oxygen requirement and angiogenesis. When we say angiogenesis, this is the formation of new blood vessels. Now, the concern here is that as the cell grows rapidly, these cells need to have proper nutrients. Okay? In a normal cell, for them to live longer, for it to grow, they need to receive proper and sufficient nutrients. But with cancer cells, let me tell you, they are hackers. Why? Because they modify themselves. Okay? As they grow bigger and bigger, they modify themselves to the fact that they won't need or they won't require many nutrient sources. They will just have to adjust to fewer amounts of nutrients. However, they also need oxygen to stay alive. And so they modify again themselves. They modify themselves again. They make these clumps of cell in your body and then they trick your body at the same time. That is why the body will produce new blood vessels that will cover these cells, that will cover them up so that they will live. The normal process or the procedure of our body's blood vessel is that they will reach every corner of our body to supply oxygen and nutrients. And so two major things that we need to remember is that normally there must be division and killing process of cells. We do not have to worry much if a cell is continuously being divided because we know that the normal process is that they will soon be killed. And that is the normal process. But if the cell continuously grow and they divide itself and the killing mechanism is faulty, it will result in a cancer cell. Okay, I've been talking about the introductory part and the important properties of cancer cells. Let us now move on and differentiate the two types of tumor. The two types of tumor. The very first type of tumor is the benign tumor. Okay? It's not B8, it's benign. Okay? Benign tumor, they are somewhat angiogenesis. I told you already that angiogenesis is the extra blood vessels that would be produced by your body so that this cancerous cell would be supplied with oxygen-rich blood. However, they are not proliferating. They do not metastasize. They do not spread into other regions of your body. They are not invasive. So meaning to say, we can treat this with, normal tumor treatment such as radiation. And after the radiation, this radiation treatment will eradicate benign tumor cells in the body. The second type of tumor cells is the malignant tumor. They proliferate, they are progressively invasive, and we cannot go against it. Why? Because they are immortal. They can spread to the other regions of your body. Now let's move on to the common types of cancer. I have stated three here. And the first common type of cancer is the carcinoma. Carcinoma is the most common type of cancer. Why? Because it is greater than 80% of cancer. It is in the epithelial tissues. Meaning to say that carcinoma can be skin cancer, can be organ cancer such as lung cancer, liver cancer, colon cancer. For most common for men is the prostate cancer. For women, breast cancer and internal tissues. Carcinoma. The second type of cancer is sarcoma. This is less than 2% of cancer. They are mesodermal tissue, so they affect your cartilage, your bone, and your fat tissue. Now, the last type of common cancer is the lymphoma and leukemia. Lymphoma and leukemia, they are about 15 to 18% directly linked with the immune system. Okay? Lymphoma, from the word suggests, cancer in our lymph nodes, like the spleen. Leukemia, it is totally different. They are single cell. They are liquid cancer. They are not in mass but floating liquid. Instead, that they are moving rapidly as a single cell but have all the properties of cancer. Now, one of the things that you will notice in the lab result of a patient with leukemia is that this patient will have a very high WBC, high white blood cells. And this is the tip of the iceberg, cancer cells. And I hope that you have learned something from this lecture, and thank you so much for listening. Once again, this is Rafter Manuel, BSN4.